Why 5 senses is wrong. For centuries, most of us have been taught that humans possess 5 senses: sight, hearing, touch, taste, and smell. This model, often attributed to the ancient Greek philosopher Aristotle in his treatise De Anima, is deeply ingrained in popular culture, forming the basis of everything from elementary school lessons to common idioms. Aristotle's framework, while groundbreaking for its time around the 4th century BCE, categorized senses based on the organs associated with them, like the eye for sight or the ear for hearing. However, from a physiological perspective, this traditional model is a vast oversimplification, failing to capture the intricate complexity of human perception. Modern science identifies distinct sensory systems not by a broad organ-centric category like touch, but by the specific types of receptors that detect stimuli and the unique neural pathways they utilize. For example, what we commonly call touch is not a singular sense but a complex interplay of multiple distinct receptor populations. These include Magnetoreceptors, which detect pressure, texture, and vibration, Thermoreceptors, responsible for sensing temperature changes, and Nociceptors, which respond to potentially damaging stimuli like extreme heat or cold or intense pressure. Each of these receptor types employs different neural pathways to send information to the brain, meaning they are functionally as distinct from each other as vision is from hearing. The Meissner corpuscles, for instance, detect light touch and vibration, while Pacinian corpuscles respond to deep pressure and vibration. Without this nuanced understanding, we miss the richness of our sensory experience and the sophisticated biological machinery underpinning it. So, while 5 senses is a convenient shorthand for everyday discourse, it masks a much richer, stranger, and far more numerous array of ways we perceive the world, with neuroscientists often counting anywhere from 9 to over 20 distinct senses. Proprioception. This is your body's hidden superpower, the sense that allows you to know where your limbs are in space without looking. Try closing your eyes and touching your nose. That's proprioception at work, guiding your hand with remarkable precision. This crucial sense comes from specialized receptors embedded within your muscles, tendons, and joints, collectively known as proprioceptors. Muscle spindles, for instance, are stretch receptors located within the belly of skeletal muscles, constantly monitoring changes in muscle length and the rate of those changes. Golgi tendon organs, found at the junction of muscles and tendons, detect changes in muscle tension. Additionally, various joint receptors provide information about joint position and movement. These receptors are constantly sending a stream of signals to your brain about their stretch, tension, and position, creating a continuous, unconscious map of your body's posture and movement. Pioneering physiologists like Sir Charles Sherrington in the early 20th century were instrumental in describing these deep sensory inputs, coining the term proprioception in his 1906 book, The Integrative Action of the Nervous System. He distinguished it from exteroception, senses detecting external stimuli, and interoception, senses detecting internal bodily states. Without proprioception, simple actions like walking would become an incredibly difficult, deliberate, and visually guided struggle, akin to learning to walk all over again. Individuals with severe proprioceptive deficits, often due to conditions like large fiber sensory neuropathy, report needing to constantly watch their feet and limbs to ensure they are moving correctly, turning every step, every reach, and every postural adjustment into a monumental and exhausting task. This highlights how fundamental proprioception is to our effortless interaction with the physical world. Kinaesthesia. While closely related to proprioception, kinesthesia is the dynamic aspect of body awareness, focusing on the movement itself rather than just static position. It's your sense of the speed, direction, and effort involved in moving your limbs. Think about catching a ball without looking, where your arm intuitively adjusts its trajectory and forced to meet the incoming object, or riding a bike and instinctively adjusting your balance and steering as you navigate a turn. That's kinesthesia providing real-time feedback on your motion, allowing for continuous fluid adjustments. While proprioception tells you that your arm is extended, kinesthesia tells you how your arm is moving, its velocity, acceleration, and the force being exerted by your muscles. This distinction is subtle but important. Proprioception informs about the state of the body, while kinesthesia informs about the process of movement. This sense is crucial for the smooth, coordinated movements we take for granted every day, from the fine motor skills required to tie your shoes or thread a needle to the complex sequences involved in playing a musical instrument or performing a dance routine. It integrates information from muscle spindles, Golgi tendon organs, and joint receptors. But critically, it also involves motor commands from the brain and the comparison of expected versus actual movement outcomes. This constant feedback loop allows us to refine and adapt our movements, making them efficient and precise. Equilibrioception. This is your sense of balance and spatial orientation, housed primarily in your inner ear within a sophisticated structure called the vestibular system. This system consists of two main parts, the three semicircular canals and the two otolith organs, the utricle and saccule. The semicircular canals are fluid-filled loops oriented in three different planes, which detect rotational movements of your head, like nodding, shaking, or tilting. When your head rotates, the fluid endolymph inside these canals lags behind due to inertia, bending tiny hair cells, stereocilia, that then send electrical signals to your brain, indicating the direction and speed of rotation. The otolith organs, on the other hand, sense linear acceleration and the pull of gravity. They contain calcium carbonate crystals called otoconia, which sit on a gelatinous membrane above another set of hair cells. When your head tilts or you accelerate linearly, like in a car, the otoconia shift, bending the hair cells and signaling the change. When you spin around rapidly and then stop, the endolymph in your semicircular canals continues to move for a moment, still signaling rotation, which creates the disorienting feeling of dizziness and vertigo. Motion sickness, for instance, is often a dramatic conflict between what your eyes see, e.g., a stationary cabin on a boat, and what your vestibular system feels, the rocking and pitching of the boat. This sensory mismatch can lead to nausea, vomiting, and disorientation. While vision and proprioception certainly contribute to our overall sense of stability, it is the vestibular system that provides the fundamental, often unconscious, sense of where up is, whether you're stable or moving, and how your head is oriented relative to gravity. Impairments to this system, such as those caused by inner ear infections or Meniere's disease, can lead to debilitating vertigo and balance issues. Interoception. This is the perception of the internal state of your body, your heartbeat, your breathing, the rumblings of your gut, your core temperature, bladder fullness, and even subtle changes in blood pressure or glucose levels. It's a crucial sense for maintaining homeostasis, the body's stable internal environment. Neuroscientist A.D. Bud Craig, in his extensive research published around 2002, particularly highlighted the role of the insula cortex of the brain in processing these visceral signals, describing it as the primary cortical representation of the body's internal state. It's why you feel your heart pound when you're nervous, the sudden pang of hunger, the subtle ache of thirst, or the urge to stretch when your muscles feel stiff. This internal awareness is also deeply connected to how we experience emotions. Some theories, like the constructed emotion framework proposed by psychologist Lisa Feldman Barrett, suggest that emotions aren't just innate, pre-programmed responses, but rather the brain's interpretation of these interoceptive signals in context. For example, a racing heart and sweaty palms might be labeled as fear if you're facing a threat, but excitement if you're about to ride a roller coaster, depending on how your brain contextualizes the internal sensation. Antonio Damasio's somatic marker hypothesis also emphasizes the role of interoceptive feedback in decision-making and emotional processing. Dysregulation of interoception is increasingly linked to various mental health conditions, including anxiety disorders, depression, eating disorders, and even autism spectrum disorder, where individuals may struggle to accurately perceive and respond to their own bodily signals. Nociception. This is the body's warning system for actual or potential tissue damage. It's crucial to understand that nociception is not the same as pain. Rather, it is the sensory input that can lead to the experience of pain. Nociceptors are specialized sensory neurons, free nerve endings found throughout the skin, muscles, joints, and internal organs that respond specifically to harmful stimuli, extreme temperatures above 45 degrees Celsius or below 5 degrees Celsius, intense mechanical pressure, like a pinch or cut, or irritating chemicals such as acids or inflammatory mediators released during injury. When you accidentally touch a hot stove, thermal nociceptors in your skin are activated, sending electrical signals via two main types of nerve fibers to your spinal cord and then to your brain. Fast-conducting A-delta fibers transmit sharp, immediate sensations, often initiating a reflex to pull your hand away, sometimes even before your conscious brain has fully registered the experience of pain. Slower-conducting C fibers transmit dull, aching, or burning sensations that persist longer. pain itself is a far more complex, subjective, and multi-dimensional experience, shaped not only by nociceptive input, but also by emotions, memories, attention, and cultural context. For example, the gate control theory of pain, proposed by Ronald Melzack and Patrick Wall in 1965, explains how non-painful input can close the gates to painful input, preventing pain sensation from traveling to the central nervous system. This helps explain why rubbing a bumped elbow can reduce the pain. Furthermore, a soldier wounded in battle might not feel pain until hours later due to the release of endorphins and intense focus on survival, demonstrating how the brain can powerfully modulate the perception of pain, even in the presence of significant nociceptive input. Nociception is the raw data of damage. Pain is the brain's full, complex, and highly individual interpretation of that data. Thermoception. Beyond just hot and cold as aspects of general touch, thermoception is your dedicated sense of temperature. You possess distinct populations of specialized receptors in your skin, mucous membranes, and internal organs specifically designed for detecting warmth and for detecting cold. These are not merely general touch receptors, they are highly specialized thermoreceptors. For instance, cold receptors, such as those expressing the TRPMA ion channel, are activated by temperatures below skin temperature, typically between 8 degrees C and 30 degrees C. Warmth receptors, often involving the TRPV3 and TRPV4 channels, respond to temperatures above skin temperature, usually between 30 degrees Celsius and 45 degrees Celsius. When temperatures exceed these ranges, e.g. below 5 degrees C or above 45 degrees C, nociceptors are activated, signaling potential tissue damage rather than just temperature. The fascinating thing is how certain chemicals can directly fool these receptors, creating thermal sensations without any actual change in temperature. Menthol, the compound found in peppermint, doesn't actually lower your body temperature, but it selectively activates the TRPM8 cold receptors, making you feel a distinct cooling sensation. Conversely, capsaicin, the pungent compound that makes chili peppers spicy, triggers your TRPV1 warmth receptors, creating a sensation of burning heat, even though no actual heat is present. This direct chemical activation highlights the remarkable specificity of these sensory pathways. Internally, thermoceptors in the hypothalamus monitor core body temperature, playing a critical role in thermoregulation and maintaining the body's internal thermal balance, triggering responses like sweating or shivering to keep temperature within a narrow, healthy range. Chronoception. This is your brain's sophisticated ability to perceive the passage of time. Unlike other senses, there's no single dedicated time organ in the body. Instead, chronoception is a complex, emergent property assembled from multiple neural timing mechanisms distributed across various brain regions. Key areas involved include the cerebellum, basal ganglia, prefrontal cortex, and even the suprachiasmatic nucleus, SCN, which governs circadian rhythms. Our perception of time is remarkably flexible and subjective, influenced by a myriad of factors. For example, time often seems to slow down during moments of intense danger or high emotion, a phenomenon sometimes called time dilation. This effect, where seconds can feel like minutes during a car accident or a frightening event, is thought to be linked to increased attentional processing and heightened emotional arousal, leading the brain to encode more information per unit of objective time. Conversely, time can feel like it speeds up while you're deeply engaged in an enjoyable activity, a state often described as flow. Or as you get older, a phenomenon known as the telescoping effect, where recent events seem closer in time. Studies, like those by Mark Wittmann and Ernst Poppel, have explored how attention and emotional states significantly distort our subjective experience of duration. This makes chronoception a deeply personal and often unsettling sense, constantly being recalibrated by our attention, emotions, memories, and even our physiological state. The brain doesn't just measure time, it actively constructs our experience of it, making it one of the most mysterious and intriguing aspects of human perception. Chemoreception in the body. Beyond the external senses of taste and smell, your body has an elaborate network of internal chemoreceptors that constantly monitor the chemical composition of your blood and cerebrospinal fluid. These are absolutely vital for maintaining homeostasis, your body's stable internal environment. Specialized cells in your carotid arteries, carotid bodies, and aortid bodies, for instance, are peripheral chemoreceptors that track oxygen and pH levels in the arterial blood. When oxygen levels drop, hypoxia, or blood becomes too acidic, These receptors send signals to the brainstem, triggering responses like increased breathing rate and heart rate to restore balance. But perhaps the most powerful and often misunderstood internal chemoreceptor system monitors carbon dioxide, CO2. Central chemoreceptors, located in the medulla oblongata of the brainstem, are exquisitely sensitive to changes in the pH of the cerebrospinal fluid, which is largely determined by the concentration of CO2 in the blood. The primary driver for the urgent, irresistible need to take a breath is not a lack of oxygen, but rather the buildup of carbon dioxide in your blood, which makes the cerebrospinal fluid more acidic. This is why some free divers can dangerously hyperventilate before a dive. By exhaling deeply and rapidly, they temporarily lower their CO2 levels, delaying the urge to breathe signal. While this extends their breath hold time, it also increases their risk of blacking out from lack of oxygen, hypoxia, without ever feeling the critical warning signal that normally prompts a breath. Other internal chemoreceptors include osmoreceptors in the hypothalamus, which detect changes in blood osmolarity and trigger thirst, and glucose receptors, which monitor blood sugar levels. This intricate internal chemical sensing network ensures that critical physiological parameters remain tightly controlled. Magnetoreception. This is the ability to detect magnetic fields, a sense well established and crucial for navigation in a wide array of animals. Migratory birds, like the European robin, famously use the Earth's weak magnetic field as a primary compass for orienting themselves during their incredible thousands of miles journeys, even in completely dark environments. Their mechanism often involves specialized photoreceptors in their eyes that are sensitive to magnetic fields through quantum effects involving cryptochromes. Similarly, sea turtles navigate vast ocean distances to their nesting beaches using both the inclination and intensity of the Earth's magnetic field as a map. Other animals, such as salmon, bats, newts, and even some insects, are known to possess magnetoreception, often employing different biophysical mechanisms, including deposits of magnetic iron minerals, magnetite, in specialized cells. For humans, however, the existence of a functional sense of magnetoreception is far more controversial. While some intriguing studies have suggested that human brainwaves can respond to changes in magnetic fields. For example, a 2019 study by researchers at the California Institute of Technology showed that alpha waves in the human brain decreased in response to specific magnetic field rotations. However, consistently replicating these findings under stringent double-blind conditions has proven extremely difficult, and the precise mechanism, if it exists, remains elusive. The scientific consensus, as of the current understanding, remains that unlike birds, turtles, or even bacteria which align themselves along magnetic fields, humans do not possess a reliable, functional, or consciously accessible sense of magnetoreception that aids in orientation or navigation. Electroreception. This is the ability to detect electric fields, a fascinating sense completely absent in humans, but absolutely crucial for survival in many other species, particularly aquatic ones. Sharks, for example, are perhaps the most famous possessors of this sense, equipped with specialized organs called ampullae of Lorenzini. These are jelly-filled pores scattered over their heads, incredibly sensitive to the faint bioelectric fields generated by the muscle contractions and nerve activity of their prey. This allows sharks to locate camouflaged fish hidden under sand even when those fish are completely out of sight and smell, providing a powerful hunting advantage in dark or murky waters. The electric fields generated by prey are often less than 1/billionth of a volt. The platypus, a unique monotreme native to Australia, also uses electroreceptors in its duck-like bill to hunt for crustaceans and insect larvae in murky water. As it swims, it sweeps its bill back and forth, detecting minute electrical impulses given off by its prey. Other animals with electroreception include various species of electric fish, like electric eels and knifish, which not only detect but also generate electric fields for navigation, communication, and stunning prey, and even some dolphins which have been found to possess electroreceptors around their rostrums. It's a prime example of a sensory modality that evolved to meet specific ecological niches, highlighting the vast diversity of perception across the animal kingdom and showing us what a truly missing sense looks like for humans, demonstrating that our own sensory world is just one of many possible realities. Synaesthesia. This is a neurological phenomenon where the stimulation of one sensory or cognitive pathway automatically and involuntarily leads to experiences in a second sensory or cognitive pathway. It's not a separate sense itself, but rather a cross-activation or blending between existing senses or cognitive concepts. For instance, a person with grapheme color synaesthesia might literally see specific colors whenever they look at numbers or letters, with the number 4 always appearing blue or the letter A appearing red. Others might experience sound-to-color synaesthesia, where different musical notes or instruments evoke distinct hues and patterns, or even sound-to-taste, where certain words or sounds produce specific flavors. There are over 80 known types of synaesthesia, including mirror touch synaesthesia, feeling touch when seeing someone else touched, and lexical gustatory synaesthesia, tasting words. It's estimated to occur in about 2 to 4% of the population, with varying degrees of intensity. The neurological basis is thought to involve increased cross-activation or reduced inhibition between brain regions that are normally more segregated, perhaps due to differences in neural connectivity or pruning during development. Composer Alexander Scriabin famously created a color organ for his symphony Prometheus, the Poem of Fire in 1910, intending for specific notes and chords to be accompanied by corresponding colored lights. A clear example of how synesthetic experiences can inspire artistic expression. Synaesthesia offers a profound glimpse into how our brains construct reality, suggesting that the boundaries between our sensory modalities are far more fluid and interconnected than we typically imagine, providing a unique window into the subjective nature of perception. Subscribe to Impulse, and the next video is waiting for you on screen right now.